Effect of different media in the growth of algae. Objective: to compare performance of NPK fertilizer and the fish meal in alga growth. What is microalgae? Microalgae are a rich source of the essential nutrients including protein, lipid, carbohydrates, vitamin, and pigment like carotenoids, making them an important component of aquaculture diet for larvae and juvenile species. As photosynthetic organisms, microalgae serve as the foundation of aquatic food webs, supporting the growth of the zooplankton and other organisms that are directly consumed by fish and shellfish in aquaculture system. Microalgae contribute to maintaining water quality by absorbing excess nutrients such as nitrogen and phosphorus, reducing the risk of eutrophication and creating a healthier environment for culture species. NPK fertilizer application in algae culture. NPK fertilizer contain nitrogen, phosphorus, and potassium. Are commonly used in algae culture to promote rapid growth in and biomass production. Nitrogen support protein synthesis, phosphorus aids in energy transfer and cellular process, while potassium enhance overall metabolic activities. Fish meal application in algae culture. Fish meal provide organic nutrients including protein and amino acids which are readily available to algae. It can stimulate algal growth and increase biomass production, leading to higher yields. The use of fish meal raises concern about overfishing and its impact on marine ecosystem. Preparation of algae starter. Prepare a nutrient-rich culture medium using sterilized water and appropriate nutrients such as NPK fertilizer or specialized algae growth media, ensuring the right balance for the target algae species. Introduce a small amount of pure algae culture inoculum into a prepared medium under sterilized conditions to prevent contamination. Place the setup in a well-lit area with a controlled temperature and aeration, ensuring adequate light exposure, natural sunlight or artificial light, and mixing to promote uniform growth. Preparation of fish meal. Collect fish processing by product, heads, bones of cut or small, low-value fish. Wash the raw material thoroughly to remove impurities like sand or dirt. Cook the fish at the 19 to 100 Celsius in a steam cooker or boiling water for 2 30 minutes to sterilize and separate oil, water, and solids. Stir during cooking to ensure even heating. Dry the pressed cake in a dryer, rotary drum, or sun drying until it reaches a moisture content of around 10%. Avoid overheating as it can reduce protein quality. Grind the dried material into a fine powder using a hammer mill or grinder to produce fish meal. Store the fish meal in an airtight bag or a container in a cool, dry place to prevent spoilage and maintain quality.
Method counting algae cells. For this method, we are going to use materials such as cover slip, hygroscytometer, algae sample, pipette dropper, Google solution, test tube, and the last one is compound microscope. Okay, for the procedure, first, prepare the materials that have been listed. Next, using the pipette dropper, take 5 milliliter of algae sample and place the sample in test tube. Then, add 1 drop of Google solution in test tube. Mix well. After that, put 1 drop of well-mixed algae suspension on each mucous chamber and cover with cover slip. Then, avoid the presence of air bubbles, overflowing, underfilling, and uneven distribution of cells. Allow the cells to settle for 5 minutes before the counting. Next is place the hygroscytometer under the microscope and observe the algae. Next, take picture of the algae at the center of the grid. And the last is then make a grid line at each square at middle of the grid. So next is then we divide the square into 9 cell sections that are symmetry. Then mark 5 cells like this diagram below. And the last one is 5 cells that have been marked is going to be count. Next is we are going to use a formula to count algae. First, count every cell that have been marked. Then add a vast amount of algae that have been marked and it will get average of cells. Then add the amount of average into this formula. Average of cell per square divided by volume of the square. And the last is the amount that from this formula is cell density and will be recorded in data collection. So this is activity that we have done. So first we take algae sample from hatchery. Then the algae sample is going to brought into the lab. First, we're going to use algae sample to mix with Lugo solution. Then the mixed algae sample is going to put on the hygroscytometer. Then we're going to observe it under microscope. And the last one, we're going to take picture of the microscope of the algae that have been observed under the microscope. So the next method is method putting media into T2 and T2 and T3. So for T2 we are going to use NPK fertilizer, and for T3 we are going to use fish meal. So for NPK fertilizer, first put NPK fertilizer into a container and put it on a scale. Second is weight the NPK fertilizer as much as 0.6 gram. And the last one is put NPK fertilizer in fish tank that have been labeled T2. And for fish meal, first put fish meal into a container and put it on scale. Second, weigh as much as 0.6 gram. And the last one, put fish meal into fish tank that have been labeled T3. So this is the activity that we've been doing at hatchery. This is bagging the fish meal, then add it into the fish tank. And this is bagging NPK fertilizer, and we are going to add it into the fish tank also.
For result on initial algal cell, week 2 show the best performance and week 1 show the poorest performance. Result on number of algae cell weekly. This graph consists of 3 different bars, which is treatment 1, treatment 2 and treatment 3, and we calculate its average value to make the graph. From the graph, treatment 2, which is NPK, 0.165 gram per tank, show the best performance compared to treatment 1 and 3. This is because nitrogen is often required in the greatest quantity by crop, primarily for vigor and yield. Nitrogen plays a key role in chlorophyll production and protein synthesis. When nitrogen is deficient, plants develop yellow or pale leaf and their growth is stunted. Phosphorus is a vital component of adenosine triphosphate, which supply the energy for many processes in the plant. Phosphorus rarely produces spectacular growth response, but is fundamental to the successful of development of all crops. Potassium is needed by virtually all crops and often in higher rate than nitrogen. Potassium regulates the plant's water content and the expansion. It has the same effect to algae because algae is under kingdom of Plantae, which means it has the same structure and function like a plant. From the graph, treatment 3, which use fish meal, show the poorest performance. It likely because the water quality are ruined by the fish meal that disturb the growth of algae.
Next is challenges. There are several challenges on culturing algae. First is lack of nutrients and light causes algae to crash. Algae need light and nutrients like nitrogen and phosphorus for growth. The bottle's design restricted light and nutrient availability, causing stress and population collapse in first experiment. Second is insect contamination. Insects like bloodworms and cockroaches disrupted algae cultures, causing infection and consuming algae. Maintaining a sterile environment and regular maintenance is crucial to prevent contamination. Third is high electricity consumption. Algae require constant light for photosynthesis. Continuous lighting increases electricity cost. Using energy-efficient systems like LEDs or natural sunlight can reduce expenses. Fourth is need for frequent monitoring. Frequent monitoring identifies issues like contamination or nutrient depletion. It is time-consuming and increases costs. Automation can help but require more investment. Next is future direction for studies. First, growth condition optimization. To increase algae biomass and nutritional content, future studies may examine strategies to better optimize environmental factors such as temperature, nutrient composition, and light intensity. Second is use of native algae strains. Studies could examine native or local algae strains that are better suited to specific conditions, thereby reducing crash risk and increasing productivity. Third is energy efficient culturing techniques. The high electricity costs related to continuous lighting can be significantly reduced by studying more energy efficient techniques such outdoor open ponds or the use of renewable energy sources. Fourth is contamination prevention. To minimize the invention of unwanted organisms like insects or competing microorganisms, future studies can focus on the development of anti-contamination methods such sterile closed culture system or biocontrol methods. Fifth is nutrient recycling systems. Studies on the systems that produce algae by recycling nutrient from aquaculture effluent could minimize costs and improve sustainability. Next is implication of the study. First, understanding algae's role in aquaculture because of its high nutritional content, especially in essential fatty acids, amino acids, and pigments. This study has shown me the essential function that algae play as a live feed for larvae organism. Second is algae culturing practical skills. The study enhanced my ability to manage algae cultures, including preparing growth medium, keep updated on growth conditions, and preventing contamination. These beneficial skills are necessary for ensuring reliability and constant feed production. Third is innovation and problem solving. Facing challenges like contamination and algae crashes during the experiment improved my ability in innovation and troubleshoot. For example, I've learned to adjust to practical challenges by adjusting the light level or cleaning the tank. Last one, broader impacts on sustainability. This study shows that algae cultivation could promote sustainable aquaculture practices. The capability to efficiently culture algae increases environmental sustainability and minimizes reliance on commercial larvae diets.
For the summary of this experiment, in this experiment, many knowledge that we got. For example, how to make fish meal, how to make starter algae, sampling algae, counting algae, select data algae, decided best treatment for algae, and how to work as a team. We also able to make this experiment running smoothly. From this knowledge that we got, we are able to culture algae effectively for fish feed.